The proteins that govern our lives move like tumbleweeds. Each has a twisted, one-of-a-kind form with prickly side branches littering its surface. If we can locate the correct key, we can defeat our most heinous foes, cancer, diabetes, infections, and even aging, which are hidden in the nooks and crannies. We recently purchased a universal key manufacturer. In a study published today in Nature, a team led by Dr. David Baker from the University of Washington developed an algorithm to design tiny protein keys that unlock those targets from scratch. Far from being an ivory tower endeavor, the algorithm took on one of the most perplexing drug development problems of our time. Can we create medications just based on the shape of a protein's lock? Welcome to today's episode of AI News. In this episode, I will show you the amazing new capabilities artificial intelligence models are gaining, which are helping us cure many diseases, among which is aging. They're not talking about any ordinary medication. Rather of focusing on tiny compounds like Tylenol, the scientists focused on protein-like molecules known as binders. While they may appear unusual, you are familiar with them. Monoclonal antibodies, for example, have been important in the treatment of severe COVID-19 patients. They're also some of our most potent anti-cancer weapons. However, these therapeutic behemoths have difficulty tunneling into cells, are difficult to create, and are sometimes prohibitively costly for general usage. What about a different approach? Can we harness the power of contemporary computation to create similar but smaller and simpler medications that are just as effective, if not more so? According to the Baker team's research, the answer is yes. The system aced its goal of screening over 500,000 possible binder structures for 12 protein targets, utilizing little computational effort compared to earlier attempts and flagging probable hits. It also discovered a cheat code that improved the efficiency with which binders grabbed onto their targets. The program, unlike earlier techniques, merely required the structure of the target protein to construct binder keys from scratch. It's a much simpler technique than past attempts. Because proteins govern our internal biological cosmos, the new software keymakers will be able to help us access the mysteries of our cells' molecular lives, and intervene when things go wrong. The capacity to produce novel proteins that bind firmly and selectively to any molecular target that you desire represents a paradigm change in drug development and molecular biology in general, Baker said. A complex network of proteins governs our bodies. Each protein bounces around the cell like a courtesan in a ballroom, briefly grasping onto another protein before moving on to the next. Specific pairings can set off cellular graphs that can either activate or block major cellular processes. Some may instruct a cell to develop or to die gently. Others may cause a cell to become malignant or senescent, releasing harmful substances and threatening neighboring cells. In other words, protein pairs are required for life to exist. They're also a strong medical hack. If any pair initiates a signaling cascade that damages a cell or tissue, we can design a doorstop molecule to physically break apart the pairing and stop the sickness. Proteins are frequently portrayed as beads on strings that crumple into complex 3D shapes. That isn't totally accurate. Protein molecular beads resemble humanoid robots, having a rigid trunk and floppy limbs known as side chains. As a protein assembles, the trunk components of its constituent amino acids are linked together to form a strong backbone. The frizz, exposed side chains, covers the protein's surface like a fuzzy ball of yarn. They generate pockets that a natural protein partner, or an imitation, can easily grip onto, depending on their location in the backbone. Previous research used these pockets to create mimic binders. However, the method is computationally demanding and frequently relies on known protein structures, a useful resource that is not always available. Another strategy is to look for hot areas on a target protein, which aren't always accessible to binders. The team approached the challenge in a manner akin to rock climbers attempting to ascend a new wall. The climbers are the binders, and the wall is the surface of the target protein. Looking up, there are a plethora of side chain and protein pocket handholds and footholds. However, the larger ones, or hotspots, may not be able to support the climber for the whole route. Another technique, according to the team, is to sketch out all of the holds, even if some appear weak. This opens up a whole new world of potential binding spots. Most will fail, but some may surprise you. A subset of these sites is then tested by thousands of climbers, each of whom is looking for a promising route. Once the top routes are identified, a second group of climbers will thoroughly investigate them. Following this parallel, the researchers added, "We designed a multi-step technique to solve earlier obstacles." To begin, the researchers searched a library of probable protein backbones as well as a vast collection of sidechain sites capable of latching onto a protein target. The original sample sizes were massive. For each target, thousands of potential protein backbone, trunks, and approximately 1 billion possible sidechain, arms, developed. The scientists refined the list to a handful of intriguing binders using Rosetta, the protein structure and function mapping software developed by Baker's team. The selection of these binders is based on conventional physics, rather than machine learning or deep learning, according to Dr. Lance Stewart, Chief Strategy and Operations Officer of the Institute for Protein Design, where Baker's lab is housed. It adds to the awe of this breakthrough. The next important question is whether or not the binders can bind in silico. But do they truly function in cells? The researchers chose 12 proteins to test its algorithm in a proof of concept. Among these were proteins with known links to cancer, diabetes, and aging. Another group concentrated on combating diseases, such as surface proteins on the flu or SARS-CoV-2. The researchers examined top candidates in E. coli bacteria after screening 15,000 to 100,000 binders for each of the protein targets. Binders were quite effective in blocking their targets, as predicted. Some block growth signals, which can lead to cancer. Others focused on a common part of influenza, the virus, which might, in principle, neutralize many strains, opening the path for a universal flu vaccination. SARS-CoV-2 was not spared, with ultra-potent binders protecting mice against its invasion. Those results were previously published. The work demonstrated that protein-like medications may be created from the ground up. All that is required is the target protein structure. The possibilities for application are unlimited, said Dr. Shores Shares, Joint Head of Structural Studies at the MRC Laboratory of Molecular Biology in Cambridge, UK, who was not involved in the work, on Twitter. The algorithm, while strong, is not without flaws. Despite the fact that millions of possible binders were discovered, only a small percentage of the designs ultimately latched onto their target. Even the finest candidates need many adjustments to their amino acid composition in order to attach to a target. However, it is groundbreaking work in a sector that has the potential to significantly revolutionize medicine. For the time being, the technology and vast dataset offer a starting point for determining how proteins interact within our cells. These data, in turn, might guide even stronger computer models in a virtuous loop, especially with a little deep learning magic thrown in. According to Stewart, it will further increase the speed and precision of design. It is work that is already under progress in our labs. So, what is your opinion on artificial intelligence quickly getting into the field of medicine? Do you think that it will continue this path, or will there be a roadblock in the near future? Please tell us your opinion in the comment section below. I would love to hear what you have to say about it. Thank you for watching AI News. We consistently report on the newest technologies that are shaping the future of our world. We'd appreciate you subscribing and watching our other videos. See you around and take care.